I would like to thank the CGD Foundation for not only funding my research project, generating recombinant human prions in large scale for structural studies, but also for organizing this virtual conference so we can share our research progress with the CGD families and our colleagues. I would like to start my presentation with some brief background introduction of the prion. The term prion was coined by Dr. Stan Prusner in early 1980s to describe the infectious agent that causes prion disease. By definition, prion is a small proteinaceous infectious particle which is resistant to inactivation by most procedures that modify nucleic acids. So basically, prion is a infectious protein. So how could a protein be infectious and cause the disease? Let's take a look at the prion protein, or PRP. It has two isoforms. The normal one, we call it a PRPC, and the disease-associated one, we call it a PRPSC. So those two isoforms have the same composition, but only differ in their shapes or structures. The high resolution three-dimensional structure of the PRPSC has been obtained for more than 2 decades. On the other hand, the high resolution structure of PRPSC is still missing, which I believe you've heard a lot from Dr. Holger-Willie's presentation earlier. But fortunately for us, there's an easy way. We can differentiate PRPSC from PRPSC by simply applying the biochemical protease digestion assay. Here on the right side, as you can see in the control normal brain, when the brain homogenate was incubated with the protease, the PRPC is chewed away or digested away. And in the CGD brain homogenate, the partially resistant PRPSC is preserved after the digestion. So we use a lot of this simple yet powerful technique to differentiate, to detect and or screen for the existence of PRPSC. So again, back to the question, how could this happen? According to the prion hypothesis, the PRPSC can interact with PRPC and convert PRPC to more PRPSC. So we've learned a lot about this process, but the detailed molecular level mechanism is largely unknown. Mostly likely it is because we are missing this high resolution structural information of the PRPSC. So the object of this project is to provide a large amount of PRPSC in its high quality, which means high purity for the structural study. So as we all know, generally speaking, for any structural study, we need the protein in its high quality, and we need a certain amount of that protein for the structural study. And here are a few ways I listed to collect or amplify the PRPSC. The first one is a traditional in vivo bioassay, which we inject an animal with different prions, and at the end of the incubation or experiment, we can get a highly infectious prion, but it is in the brain. We need to purify it, and the experiments typically last for months, if it's not years. So it's going to be time consuming and cost a lot. The second and the third ways are the in vitro based way. And in the second way, we still use the brain homogenate, but we don't need to house the animal. We just take the animal brain, homogenate that brain's tissue, and use that as a substrate to amplify. The resulting PRPSC is highly infectious. We can get multiple strains, but again, we need to purify it. But compared to the first method, it's time and cost efficient. The third one, instead of using the brain homogenate, we actually purify the recombinant PRPC from bacteria to start with, and the resulting PRP-SC would be relatively pure. We don't need to purify, or even if we do need, it's a simple step. And of course, we can tell it's time and cost efficient, but the only uncertainty is the infectivity and the stream property. We need to verify those. So here, I'm gonna tell you using this method how I amplify the recombinant prion by PMCA, or protein misfolding cyclic amplification, which is the technique that Dr. Soto invented about 2 decades ago. So the idea is PRP-SC has the seeding ability, which when you mix the PRP-SC and PRP-C together, PRP-SC would induce the conversion to generate more PRP-SC, and we simply apply the mechanical force to break down those newly formed PRP-SC to generate smaller aggregates. Then if we provide enough PRP-C substrate with the cycles of incubation and sonication, we would be able to see the production of endless PRP-SC. That's the principle. Here's how we do it in our lab. Here's the sonicator. Here's the PCR tube we typically use. In the PCR tube, we add like 90 microliter typically of the substrate. Then we add a tiny amount of the seed into the tube. And after cycles of incubation and sonication, those PRP-C in the tube will become PRP-SC. And then we can take a little amount from this tube and transfer to another tube to seed the reaction again and again. So like I said, as long as we provide the PRP-C substrate, we would be able to indefinitely continue this amplification. Here's the condition I use to generate or propagate the recombinant prion using the mouse recombinant PRP. The results are here. As you can see, we were able to generate those protease-resistant recombinant prion. Of course, I need to prove you that those are indeed prions. So the first thing we did was to try to look under the transmission electron microscope to compare the morphology between our newly amplified recombinant prion and the native RML prions that purified from diseased mouse brains. As you can see, from my eyes, I would say they look very similar, but we need to make sure they are indeed behave similar, biologically speaking. So we inject those recombinant prion into wild type animal, 8 of them. All of them developed a prion disease within 140 days, and they died around 160 to 170 days, and the average survival time is 163 days. And here's the survival curve. All the control animals are still healthy, 400 days, more than 400 days after inoculation. The next is the biochemical characterization. This image or the experiment tell us that the mobility of the PRP-SC after protease digestion is exactly the same as the native RML prion, and the glycosylation pattern is the same as well. Those two aspects strongly suggest to us the recombinant prion we generated or propagated in vitro by PMCA is indeed a RML strain. And here's another supporting evidence is the serial protease digestion profile. Those two curves between the native RML and recombinant RML are very close. Of course, there's another very important comparison study, which is the lesion profile study. It is ongoing. We hope to have it by the end of next month, but we're very confident that the recombinant prion is indeed a RML strain. So here I'm showing you a cryo-EM image taken by one of my colleagues in our university. using our newly established cryo-EM facility. We're very excited to to generate more recombinant RML Prion and to put on the microscope, try to obtain the high resolution structure information of the RML prions. So back to our object of this project, it is to generate recombinant human prions, which I used the similar setup using the recombinant human PRP as a substrate and various of human prion, native prions as the seed, such as the variant CGD. I tested first. I've tried multiple conditions. Unfortunately, so far, all of them came back negative, which means I didn't able to detect any protease-resistant recombinant PRP, which is very disappointing, but that's what we sort of expected because it is hard to work with human prion. But there are a couple of things we definitely can improve. One is, of course, we can tweak the conditions more. We try more different conditions. The other one is we only use the variant CGD in our trial experiments because simply we have large amount of supply of the variant CGD brain homogenated. We can afford to test the different conditions. As you can see, each time in those experiments, we use at least 10 microliters. So we have tons of the variant CGD, so we wanted to sort of like test the conditions which can support the amplification of human recombinant prion. That being said, we also know that sporadic CGD, when we amplify it using the PMCA, not the recombinant, but the brain homogenate from transgenic mouse, the conditions are different for variant and sporadic CGDs. So we're still optimistic. We can try different conditions, different seeds, and we're still optimistic that we can make it work. That being said, we cannot just put all the eggs into the same basket. So right now, this is still ongoing, but not working. We're looking at other options. The first one, we can do it, but it's going to take longer time and a lot of money. So we're going to rely on the second one, the one going to use the brain homogenate from the transgenic mouse as the substrate. So here is the traditional PMCA we tested, and we optimized the condition to amplify the human prions. Those are the sporadic CGD MM1 type, MM2 type, and the VV2 type. They're on the different substrate. The M substrate at 129 or the valine 129 substrate. One thing I want to point out, those experiments were done in the PCR tube, which has the capacity of 200 microliter. Usually our experiment was carried out in 100 microliter. It's a tiny amount. So to better facilitate the structure study, we need a larger amount. So we want to bump up the scale. So the idea of larger scale PMC is to use larger container. Here, I'm showing you a cup which has the capacity of 20 ml. And with this 3D printed cup holder, we can put 7 cups at the same time. And the maximum volume would be 20 ml. So, but I started this large scale format with RML, Prion, and a wild type mouse brain homogenate as a substrate. It's simply because they're relatively easy to get, and we can afford the failure. But fortunately, the first experiment, the first cup worked very well, as it is very similar to what we seen with the PCR tube PMCA format. So to make sure this is not a fluke, I tried tested other cups, like 3 more cups at the same time, and they all worked very well. So we're confident this larger scale PMCA format is a robust robust system. So then we applied this to the human prions. The good news is they all worked, at least for the sporadic CGD VV2 subtype. Here is the inoculum or the seed, and within 2 days, it has reached its amplification plateau. And the same is the variant CGD on the methionine 129 substrate. Within a week, less than a week, day 6, it has reached its plateau. So here, typically in this cup, we use 5 ml of the brain homogenate. 5 ml brain homogenate is equal to 1 mouse brain. So if you consider this is 1 mouse instead of in the cage, now in the cup, within a week, we can collect as much of the PRP-SC as we would be able to collect in the mouse, living mouse, but we need, in the cage, we need to wait for months. So obviously, this format is much more time and cost efficient. So the next step would be try other subtypes. We're very confident they all would work. Also bump up even this from this one to an even larger cup. We have tested a larger cup with a capacity of 0.5 liter on the recombinant prion, and it worked. So we're very confident with this larger scale PMCA format, we would be able to provide enough human prions. And the only thing we need to do is to purify it out of those reactions, which we know there's now a relatively easy way. It was published by Dr. Wadley's group a few years back. So to summarize the study, I hope my data shown here would convince you that recombinant prion can be generated using the recombinant PRP as a substrate, and inoculate the native prion as the seed, and the amplified recombinant prion actually indeed preserves the inoculum original prion string properties. Also, we are confident that the large-scale PMCA format is ready to produce massive amount of PRPC, either in the recombinant form or the endogenous form, which we believe would facilitate the biochemical, biophysical, and structural studies of all the infectious prions. To conclude my presentation, I would like to thank again the CGD Foundation for funding my research project. Of course, all the families who have contributed to the grant, which made this project possible. In the end, I'd also like to thank my colleagues for having supported and helped and collaborated with me to move this project forward. And thank you very much for your attention.